This material is made available to you by or on behalf of the University of Melbourne under Section 113P of the Copyright Act 1968. It may be subject to copyright. For more information, visit the University Copyright website.
All right, I'm just going to start recording now. Thank you very much, everyone, for joining today's seminar. It's a really pleasure to introduce our colleague, Peter Irwin. Excuse me. Peter, I know Peter for almost last, I would say, 10 years, when I first met with him in Perth at his place for a cup of tea with Ian Beveridge. So I still remember that. Peter is a small animal medicine specialist, has also worked at Werribee. Some of you know already him, I think. And then Peter formally retired as a professor from, and also head of school from Murdoch Uni. And his passion is on vector-borne diseases of companion animals. And today he's going to talk about some of his work on emerging tick-borne diseases of companion animals. Thank you, Peter, for joining. Over to you.
Thank you very much. Lovely to be with you. Good afternoon, everybody. Now, can you hear me okay?
Yes, we can hear you. Thank you.
Good, good. All right, let's see if I can get the technology to work. We didn't test the slides that they could actually advance, did we?
That should be fine.
Oh, there we go. There we go. Okay, well, yes, thanks for the introduction. And as you say, you know, ticks and tick-borne disease have been a passion of mine for a long time. So it's really good to be able to have this opportunity to talk a little bit about them. I'm going to focus particularly during this talk on the latest disease to emerge in Australia. That's canine monocytic ehrlichiosis. But I thought I would just start by giving a little bit of an overview of ticks and dogs in Australia. I'm not quite sure. I think we have a mixed audience, and so just a few words about ticks by way of background. And I'll talk a little bit about the major tick-borne diseases of dogs. I'm only talking about canine diseases today. And then, as I say, focus on ehrlichiosis and of another recently discovered infection. But if you thought you were just going to be able to sit back and drink a cup of tea or coffee or gin and tonic, what time is it in Melbourne? Probably time for a glass of something.
It's a lunchtime.
Oh, yes. Well, maybe you shouldn't be drinking anything other than a tea or a coffee. But I've got a quiz for you here. So which of these diseases, canine diseases, tick-associated diseases of dogs, do you think is the odd one out, if you like, or the exception? But more importantly, why? So the diseases are listed there on the left-hand side: Babesiosis, Ehrlichiosis, the Haemotropic Mycoplasmas, Anaplasmosis, tick paralysis, and hepatozoonosis. And I'll let you think about that for a few minutes while I just progress with a little bit of introduction about ticks. So, as I'm sure most of you know, these are arachnids. They're not insects, and the suborder Ixodida is divided into two main families: the hard ticks, the Ixodidae, and the Argacidae, the soft ticks. And they comprise the vast majority. There's just one outlier, the Nataliidae, one species of tick that happens to fall into this family. But for the most part, the hard ticks and soft ticks are the ones that concern us. And to a large extent, it's the hard ticks as a group that are most significant in terms of pathogenicity and disease transmission. Now, within this large group of ticks, there are approximately 69 species that are native to Australia, which is quite a sizable proportion, actually. And then in addition to that, there are 5 species of tick introduced into Australia, and I'll talk more about that as we go along. So ticks are blood-feeding ectoparasites. They live on the skin. During the course of them biting and feeding, they transmit pathogens, and they trigger immune responses in the hosts from which they're feeding. And they have a wide range of vertebrate hosts. These are usually wildlife, and therefore companion animals or domesticated animals and people can become infected when they encroach into the wildlife tick habitat. But there are a few species of tick that have become adapted to domestic animals, and these themselves cause economic and emotional cost when they cause disease. Ticks have 4 life stages: eggs, larvae, nymphs, and adults. And most ticks are what are referred to as 3-host. In other words, each of the main stages, the larvae, the nymphs, or the adults, feed and then drop off the host into the environment, molt, and then seek another host. So during that molting period, when the tick is in the environment, they're particularly susceptible to environmental conditions, but also it means that the total life cycle from egg to adult may take a very long time, may take years, depending on the climatic conditions. When they're seeking a new host, they have several different tactics, sometimes referred to as questing or ambush, but whichever way they do it, they look for their host. This is a very short video. This is not an Australian tick. This is a North American tick, but just to remind you, they scuttle around like this, and this is a questing tick, looking for a passing animal. So as I said, there are quite a few, nearly 70 indigenous native tick species, comprising four genera. And they've evolved like our unique wildlife in relative isolation. So phylogenetically, they're pretty distinct. And for the most part, these 69 or 70 species of tick occur only in Australia, some in New Guinea, and then there are a few, for example, ticks of seabirds that do travel from the southern hemisphere to the northern hemisphere, and where those traveling seabirds go. But a take-home message that I think is really important for understanding tick-borne diseases, such as we understand them in Australia, is that the ticks here are very different to ticks that have been studied elsewhere in the world, particularly in the northern hemisphere. We do not, for example, have our own native species of Rhipicephalus, Dermacentor, or Hyaloma, and the first two of those genera are certainly very important for disease transmission overseas. So there have been 5 species of tick then introduced with domestic animals, and for the most part, as far as we understand, these have come in the last 232 years, whatever it is, since European settlement. And you can see on the right here some images of the introduced animals they came with. And I say introduced with domestic animals, well, possibly shouldn't really call a fox or a rat a domestic animal, but the point is that these ticks came in with those hosts and were imported into Australia. So I come back to the question that I posed a few moments ago: which of these tick-associated diseases is the exception, and importantly, why? Well, hopefully you all got full marks by realizing that tick paralysis is the odd one out here, if you like. And I think there are 2 reasons. The point I really want to make is that tick paralysis is the only disease that's caused by native or indigenous tick species, and it's caused by the paralysis tick, the eastern paralysis tick, Ixodes holocycus, and cornuatus. the other answer that you could have given, of course, is that, well, tick paralysis is not an infectious disease. It's caused by a toxin, whereas the other 5 are infectious diseases. So I'll pay all of those answers. But all of the other 5 diseases here are transmitted by the ticks that were introduced with the domestic animals. And the one that I'm going to concentrate on, because it is by far and away the most important, is the brown dog tick. It's an extremely effective vector of disease. These are images taken in Townsville, the top 2 images of quite a heavy burden of ticks in the ear and the feet. These are the predilection sites for the brown dog tick. The image on the right there, at the bottom right, shows males and females and one little nymph. The brown dog tick actually has had quite a makeover in recent years and has been redescribed using both morphological characters and genetic markers. For many years, I think we all thought that the brown dog tick was just the brown dog tick, regardless of whereabouts in the world it occurred. However, important, I guess, for this discussion as well, is that it was known that in certain parts of the world, particularly Europe, for example, one of the major diseases transmitted by the brown dog tick is canine Ehrlichiosis. Well, canine Ehrlichiosis is quite well known, quite common, important disease in the eastern part of the Mediterranean, around Israel, Greece, and Egypt, but rarely occurs in Spain, Italy, for example, less common in Italy. And yet, despite that, the brown dog tick is prevalent throughout the whole area. And this began to raise questions about whether the brown dog tick really was just the brown dog tick, or whether there were different subspecies. And over time, both, as I say, morphologically and molecularly, it's been found that there are indeed 2 distinct lineages of this tick, referred to as the temperate lineage and the tropical lineage, sometimes also known as the southern and northern lineages. And those 2 terms actually come from a South American perspective. So the southern lineage is the more cold, temperate lineage, whereas the tropical is the northern parts of South America, of course, are in the tropics. And as if that's not confusing enough, the brown dog tick, Rhipicephalus sanguineus, has also undergone a name change and has recently been renamed as Rhipicephalus linnaei. Now, what's all this got to do with my talk today? Well, it would be important to know which type of Rhipicephalus we have in Australia, since the lineage is important with respect to vectorial capacity. And to date, there's no evidence of the Rhipicephalus sanguineus sensu strictu, that's the temperate lineage here. All the work would go to show that it's the tropical lineage. And this is interesting not only for people who like ticks, but also it raises the question, when did this tick arrive and how did this tick arrive? Because I think it was widely assumed it had probably come in the last 250 odd years, 230 years, again with European settlement. But the dingo was the only large placental carnivore on the continent for a very long time. And it is a dog that will certainly carry these ticks. And so the question now is that if we do have the tropical lineage here, maybe this did come with the dingo and has been here for much longer than originally thought. Be interesting to see if anybody listening today has any further insights on that. So the range of the brown dog tick in Australia, Rhipicephalus linnaei, is broadly tropical and subtropical. There have been a number of studies over the last few years. We've done some from Murdoch University, and some have been done also out of Sydney. And the image on the right gives a a sort of rough idea, if you like, of where this tick can be found. So extending, throughout all of the Northern Territory in Queensland, but extending into northern New South Wales, northern parts of South Australia, and through tropical northern WA and down the coast. But the problem with a map like this, of course, is that there'll be many other places, certainly in the more northern regions, where it occurs, yet don't fit within that area. So moving on now to the companion animal tick-borne diseases, the infectious diseases. This is an overview with a timeline of their occurrence and their development over the last 20 or 30 years. And you can see here that PCR became the technique that has greatly improved our ability to detect many of these organisms, because generally they are subclinical. They're sometimes referred to as stealth organisms for the large part, and PCR has greatly enhanced our ability to detect it. And in fact, if I take out the Mycoplasma canis that was found in 1971, but really not seen again for the best part of 30 or 40 years, you can see this very large gap that occurred between the original discovery of the first canine vector-borne disease, which was Babesia vogeli. Back then it was called Babesia canis, and then Anaplasma platys. But PCR has greatly enhanced our ability to find some of these organisms, and indeed, as I'll explain a little bit later, some have been discovered only by PCR initially before being seen microscopically. So the first disorder, I'm just going to run through very quickly, give you a very short explanation or overview of each of these diseases. Canine Babesiosis was the first that was discovered here in Australia, 1966, by Hill and Bolton up in the northern tropics. And it's transmitted by the brown dog tick, as are all of these. And it's considered by world standards a relatively mild Babesia pathogen. There are others, such as in South Africa, and the European species are very much more pathogenic. But nevertheless, this is a serious infection in young pups up to an age of about 12 weeks old, in which it causes severe anemia and is fatal in some cases. The organisms there, red blood cell parasite lives in the red blood cells. Interestingly, the second Babesia was the next cab off the rank, and this organism has been certainly discovered, within Australia, particularly in Victoria and the East Coast. And whereas it can be and is transmitted by ticks in other parts of the world, this organism has gained some degree of notoriety as being associated with blood transmission during fighting. So it has been seen sometimes as a proxy for discovering dogs that might have been involved in fighting. Not necessarily illegal type dog fighting, but it's been found in some dogs that have normal dogs, or as I say, dogs that are not involved in illegal fighting, having been bitten by dogs that are carrying this infection. So it's been transmitted around the world in certain breeds of dogs. The dogs that are used for fighting, like American Staffordshire Bull Terriers, Pit Bull Terriers, and the like. And of course, in most parts of the world, that practice is banned, as it should be. But it's a difficult disease to study because in most countries the owners of such dogs are not very willing to submit their dogs to study, for obvious reasons. But we do have Babesia gibsoni infection in Australia, but not very commonly reported. And then there are other bacteria. There are bacterial infections, Haemotropic Mycoplasma, for example. This is the very first one that was discovered. Again, it was called something different back in 1971. It's called Haemotropic canis back then. The microbiologists have been at it, and it's been renamed over the years to now being Mycoplasma haemocanis. It also isn't very pathogenic, but in dogs that either become immunocompromised through a cancer treatment, for example, or particularly if they are splenectomized, have their spleen removed, then this can cause a hemolytic anemia. And there are several different species of these Haemotropic Mycoplasma. These are bacteria, mostly transmitted again by the brown dog tick, as opposed to the feline Haemotropic Mycoplasma, which is transmitted by fleas. But we have a number of different species of these organisms now recognized in Australia. Way back in 2001, another bacterial tick-transmitted disease was identified. This is anaplasmosis, and it caused quite a lot of excitement at the time because, again, back then it had a different name. This was originally referred to as Ehrlichia platis. And so that editorial that was written in 2001 got everybody terribly excited, referring to canine ehrlichiosis in Australia. Well, indeed, back then it was an ehrlichiosis, not canine monocytic ehrlichiosis, not the really nasty one. But this is Anaplasma platis, which is another relatively mild pathogen, but very common throughout the north of Australia. And as I say, its name has changed over the years. Chronologically, the next organism to be identified was Hepatozoon canis, but I'll come back to that in a moment because I want to spend the next 10 minutes just talking about Ehrlichia and Ehrlichiosis, which is the most serious tick-borne disease of all of these I'm speaking about, and the most recent one to arrive in Australia. So, as I expect most people know, this was originally identified in Kununurra, northern WA, in 2020, in April, May. That seems a long time ago now, doesn't it? And it has, over the last 2 years, become endemic, certainly throughout the Northern Territory and northern WA, and is rapidly spreading across North Queensland. It's considered to be well established now in the Mount Isa region and in communities north of Mount Isa and in the southern Gulf country there. Since its first identification, we've had at least well over 1,000 cases reported. It's a notifiable disease as well in Australia. So vets are obliged to report suspected cases and have it confirmed. The disease, this is a disease of canids generally, domestic dogs particularly, but also wild canids, and it's caused by a bacterial infection. Ehrlichia canis, this is a form of rickettsial organism that once it gets into the blood after having been transmitted by the tick, makes its way to a certain group of cells, the monocytes and macrophages, where it multiplies. It's an intracellular organism, multiplies in these groups and causes inclusions like you can see in this image there. It becomes widely disseminated through the animal's body and induces massive antibody response, but the antibodies for the large part are non-protective and troublesome insofar as they cause unwanted pathology such as immune complex formation, which in turn leads to widespread inflammation, which underpins the disease severity and the clinical signs. The transmission of Ehrlichia canis is quite rapid compared, for example, to Babesia, which takes several days to be transmitted by an engorging tick. E. canis is transmitted within about 3 hours of the tick attaching. And that's a whole new paradigm for vets in Australia because of how we then protect dogs against this disease. It's also transmitted mechanically by male ticks. So male Rhipicephalus sanguinis, male brown dog ticks, have a habit of traveling quite fast around the surface of a dog's body and looking for female ticks, and also will quite easily and willingly hop. They don't hop, but they move from one dog to another quite rapidly in search of females. And as they go, they feed a little bit here and feed a little bit there. They don't engorge in the same way as a female tick does, but they take small bites of blood. And it has been shown that male ticks can transmit this infection mechanically from one dog to another by feeding on an infected dog and moving to an uninfected dog. In addition to that, co-transmission is very common because the brown dog tick is the vector tick. I've already shown you a list of a number of other agents that are transmitted by this tick, and so co-infection is often seen with Ehrlichia canis. The whole genome has been analyzed, of Ehrlichia canis has been analyzed, and it's been shown that the strain, if you like, of E. canis that we have here in Australia is referred to as the Taiwan genotype that is common throughout Asia and the Middle East. And the study that was done by Neve et al. Out of CSIRO in Geelong have shown that all the isolates that they studied throughout quite large geographical areas, so the Northern Territory, Northern Western Australia, South Australia, all the isolates were identical, which perhaps would be consistent with a recent introduction, as we suspect, and a rapid spread of that isolate. However, what is not known is the exact time, date, and even the location of the original incursion. As I said earlier, the first cases were diagnosed in Kununurra, but it's not known whether that part of Western Australia was where it first occurred, where it first came into the country. It is a really serious disease of dogs, and the reason for that is that it has fairly, certainly within Australia at the moment, unique epidemiological features. However, as I think I said just now, the immune response to E. canis is non-productive and in fact counterproductive. So we get a whole load of pathology associated with infection, unwanted pathology. And treatment is also, unfortunately, not 100% effective. The disease has had an absolutely devastating effect on dogs within Indigenous communities and the Indigenous communities more broadly, where very high tick burdens are evident, and there have been mainly anecdotal reports of severe morbidity and mortality, so many sick dogs and high mortalities in those communities, and associated with this rapid spread across the top end. And if you like, it's been a perfect storm, I believe. We have had for many years a very well established tick population throughout Northern Australia. And remember, this is Rhipicephalus linnaei, or the tropical lineage, which is an extremely good vector for Ehrlichia canis. So it's all, the ticks have just been waiting for this to happen, if you like. And the climatic conditions in the north are perfect for the tick, and in Indigenous communities, tick control has been suboptimal, I guess. And so, as you saw from those pictures I showed a few minutes ago, some dogs can have very heavy burdens of ticks. But more importantly, this is a tick that is adapted to dwellings, and so within the buildings, within the kennels, within people's homes in Northern Australia, there are often quite large numbers of ticks. So we have this large population of ticks. We have a naive dog population that's naive with respect to Ehrlichia, and in comes Ehrlichia. So we've had this epidemic, if you like, with high casualties. And speaking of high casualties, this is analogous, I believe, to the situation in Southeast Asia in the mainly 60s and 70s, where many military working dogs, generally German Shepherds, were brought into that part of the world as part of the Indochina War, Vietnam War, and there were enormous numbers of casualties, deaths amongst these military dogs. And it was a similar situation with differences, if you like. There was an established tick population, but those ticks carried Ehrlichia canis, and into this, into rural Vietnam came this population of naive American, British, Australian military working dogs. And as I say, there was enormous casualties. And it's really quite instructive to read back on those original papers in the late 1960s and during the 1970s. Initially, it really wasn't known why their dogs were dying. These were important for military purposes, I guess. And there was a high mortality amongst these dogs. And if you read the clinical descriptions at the time, there are very great similarities with what's been seen in the Aboriginal communities and across northern Australia over the last couple of years. This isn't really a talk about clinical disease, but I think what will happen over time is, as the dog population increases its immunity and exposure to the parasite, to the bacterium, it will probably develop into a situation like in other parts of the world now, where it's always been a serious disease, but there's a certain endemic stability. The clinical signs of Ehrlichia can be quite severe, as I've discussed, but generally also quite non-specific. So the dogs could be just generally unwell, lethargic, losing weight. They usually have quite a high fever, and then there could be a number of other slightly more specific signs or very obvious signs. Eye disease, they become pale, they become anemic, they become pale, and very very obviously and distressingly for the owners, they get bleeding problems. So they bleed from their noses. And I have a series of pictures here for you, taken by colleagues in the northern parts of Australia, mainly through AMRIC, wonderful organisation AMRIC, and also by John Beadle in Broome, who's a vet in Broome. You can see here dogs with eye problems, these cloudy eyes. The dogs with bleeding issues, they can bleed profusely from their noses, epistaxis, so much so that they can die of blood loss from hemorrhage. But they also become edematous, swelling. They get swelling of their neck, their general body, the scrotum, interestingly. And again, that's as a clinical sign, was reported widely during the Vietnam conflict and the vets who were treating those dogs, but has hardly ever been seen or reported since. I just find it interesting, this similarity that we find ourselves in now compared to back then. The dogs become very skinny. They're very susceptible to infection. The dog on the left there had just got a cut in its foot because its bone marrow was impacted. It was unable to mount an immune response. That dog died of sepsis as a consequence of the infection. As I said a few minutes ago, it is a notifiable disease. So currently, I expect that will change fairly soon. Now it's becoming endemic. But until now, and including now, it is mandated that vets test suspected cases. But the challenge is that we don't have a very good, what I'd call cage side or in clinic test available at the moment. And hopefully that'll be something that research will provide vets with a good test in coming months to years. The disease is treatable with antibiotics. So the drug that's used is doxycycline. However, it's not 100% effective. And a small number of dogs, for whatever reason, the organism survives in treated dogs and can persist. So it really becomes very important to prevent infection. And the very rapid transmission of this pathogen from the brown dog tick necessitates a strategy that's a little bit different to the main strategies, for example, that are used for paralysis tick. And I'll explain as we go. So there's a newish group of drugs, the isoxazolines or the isoxazolines, which are really excellent acaricides. They kill ticks very efficiently. However, they work systemically, so the tick has to be attached and has to be feeding on the dog in order to be killed. And that dynamic is too slow for the prevention of the transmission of E. canis, because that's transmitted within 3 hours. So specifically for this infection and this disease, you need a drug or a compound that is repellent, has a repellency action and kills the ticks before they bite. And the drugs that contain, the products that contain flumethrin, for example, or permethrin, have that property. So in communities managing outbreaks, the collar, so Seresto is the only product that is licensed for the prevention of ehrlichiosis in Australia, should be used with an isoxazolin. But in a positive dog, to prevent onward transmission, an isoxazolin on its own would be fine. But it really has, I guess, challenged vets' thinking about how to protect dogs against diseases transmitted by ticks. So vets in practice have had to revisit their parasitology, which of course is a really good thing. Okay, just finally, before we wind up, I wanted to tell you one more sort of interesting story. I think it's interesting about one of the last tick-borne diseases that's been discovered here. This is hepatozoon canis. I have a personal sort of interest in this because my PhD student at the time made this discovery, and it sort of illustrates, I think, how nationwide surveys now based molecularly with PCR, can be very useful and turn up things you don't expect. So a few years ago, 5-6 years ago, we were doing a nationwide study of ticks in Australia, a tick survey, if you like. And we were collecting ticks from all around the country, so vets and anybody, dog owners were collecting ticks for us. And we published all this work, of course. But one of the ticks that was taken one day off a little dog, this beautiful little puppy here, is a Maremma sheepdog. This was in Serena in mid north coast Queensland. This little dog had tick paralysis. Fortunately, wasn't too unwell with it, was treated appropriately and survived. But the tick that was causing the tick paralysis tick, Ixodes holocyclus, was removed and submitted for our research. We eventually got around to analyzing what was in those ticks, and to our great surprise, we found hepatozoon canis. And again, I like this this example or this little story because it combines the new and the old. So we discovered it unseen by PCR. And because we had good records, we were able to contact the vet and go back and eventually find the owner of this individual dog, who was a very nice lady, and the dog was still alive. It had survived from its tick paralysis. It was now a couple of years later, a very fine Maremma sheepdog. And we got permission to take a blood sample from the dog. We looked in its blood, and sure enough, sitting in its blood 2 or 3 years later was the organism. I don't know if you can see the arrow here, but you can see the organism within a white blood cell. So that's Hepatozoon canis, and it made a nice story. We'd confirmed it molecularly, and then we could actually see it in the dog's blood. So Hepatozoon canis is, as you might suspect from the story, not a very pathogenic organism. It's another parasite of canids, but particularly foxes. So it's an organism that is transmitted between foxes by ticks, usually the ingestion of the tick rather than the biting of the tick, but it can also be transmitted by predation and vertically from mother to offspring. What's really interesting about Hepatozoon canis is that it's not aligned with the tropical lineage of Rhipicephalus sanguinis. So it's not aligned with the type of brown dog tick that we have here. And so we sort of come full circle to what I was talking about earlier on. The question really is, how did the Hepatozoon canis come in? Well, we can say it's unlikely with the brown dog ticks because we know that the brown dog tick in Australia is the tropical lineage, and we know that Hepatozoon canis is not generally transmitted or associated with that lineage. And therefore, it's probably unlikely that it came in with the dingoes originally. So we presume that Hepatozoon canis came in with dogs rather than ticks, imported into Australia at some stage in the last 230 years. It's possible that it came in with the foxes. I expect there are some people listening to this saying, yes, well, of course it came in with the foxes, not the dogs, because they're the main host. But I think possibly for this little pup that had Hepatozoon canis, it probably came from its mother, where the mother, so it was a vertical transmission to the pup, almost certainly, because it was a very young dog at the time, hadn't had any tick exposure apart from the paralysis tick that was just drinking its blood and acting as an amplifier for our studies. But I think that pup got it from its mother, but where the mother got it from, I don't know. We did try and trace family histories and all that sort of thing, but we're unsuccessful in that quest. Well, that brings me to the end of my presentation. I hope that I haven't bored you too much, and I hope that that's been of interest. I would just like to acknowledge and thank the many people, particularly with regard to Ehrlichia and the Ehrlichiosis outbreak, who've been involved in our studies, been involved in collecting samples, who've been involved in informing me about it, who've been taking photographs of the dogs, and a particular shout out to AMRIC, Animal Management in Rural and Remote Indigenous Communities. Just a simply wonderful organization, but also other vets in the field, and to Mark Shipp and his department as well. So with that, I'll thank you very much and stop sharing.
Thank you very much, Peter. Questions for Peter? While people are thinking about question, Peter, can I just ask that vets working in Victoria, what sort of things, I mean, although you have touched on certain things, you know, as a practicing veterinarian, and you have also worked in Victoria as well. So what sort of things now people should be considering in their, you know, kind of list of different diagnostics in terms of vector-borne diseases in companion animals?
Sure. I deliberately didn't want to make this too much of a clinical talk, but the question's a really good one, and that is that I guess vets, one of the really important pieces of history we always try and collect is about travel history. So for Victorian vets, they are most likely to encounter dogs with Ehrlichiosis if the dog has travel history from Northern Australia. So don't just think that this is a dog that's been living in Darwin and come to Melbourne. We have people, especially in post-COVID times, more and more people again are beginning to travel. So we talk about grey nomads, but travelers around Australia going up north, taking their pets with them, taking their dog with them, and then coming back to the southern states. But in addition to that, there are organizations that rehome dogs. So rehoming organizations, they're very well aware of Ehrlichia, and of course are doing what they can to prevent transmission or prevent spread. But the first part of the answer to your question is the travel history is critical. If you can find out if the dog's been up north and you've got a patient with clinical signs, clinical symptoms, if you like, that are consistent with Ehrlichiosis, then always try and find out more about its travel history. After that, those clinical signs, are what you look out for, and then with some good problem-oriented medicine and approach to the problems, ruling things out and ruling things in is the way to go. But dogs that are febrile have bleeding problems, so epistaxis, bleeding from the nose, or petechial hemorrhages, dogs that have low red blood cells, white blood cells, platelets in particular, those sorts of hematological abnormalities should raise the suspicion of Ehrlichiosis. And I've just seen, I can now just see many people who are listening in, which is fantastic. And I can see Amric's there. So hi, Amric.
Marshall has raised his hand. We'll come back to you, Marshall. There's a question in the chat that can you comment on any human transmission, please? Anna is asking.
Okay, thanks, Anna. Overseas, it has been known that Ehrlichia canis can infect people. These cases have only been reported in South America, and a very small number of cases at that. But there were a couple of really quite good studies 10, 15 years ago to show a small number of people had the South American strain, if you like, of Ehrlichia canis. So there is a zoonotic association. However, the strain, as I said, was South American, not the Taiwan genotype. But so I think it's very unlikely that the Ehrlichia canis currently in Australia has zoonotic implications or risk to people. But there's a lot we don't know about vector-borne diseases, so it is something that I think deserves some research in its own right. But I think the risk, knowing that the phenotype, the genotypic makeup of the organism is very low.
Marshall has a question.
Marshall.
Absolutely fabulous talk. Thank you very much. Enjoyed it immensely.
Thanks,
What's your others' predictions for the impact on dingoes?
Yeah, look, that's a really good question, and potentially really worrying, isn't it? I know that anecdotally, and I mean, you've had a lot more to do with dingoes, I'm sure, than me, but anecdotally, they're a breed, a creature that seems to be relatively resistant to ticks. I'm hesitant to say that, but because brown dog ticks have been found on dingoes, of course, but they don't seem to be tremendously good hosts for the brown dog tick. But that said, they're a dog, and they theoretically are going to be susceptible. I think one of the big problems with the domestic dog situation is the very, very large numbers of ticks in small confined areas, like we're seeing in indigenous communities in the north at the moment. So it's sort of the transmission is there's a very high infectious load, very high transmission rate to the domestic dogs, and I suspect that won't repeat itself very well within the dingo community. But the brown dog tick can be considered almost like a nidicolous tick, a nest associated tick. It survives so well, in sheltered areas around dogs, which is why it does so well in homes. So I think there is potential risk for the dingo breed. I don't know, perhaps others who are listening might have other ideas or other comments or other experience. I haven't yet heard of a dingo with ehrlichiosis, but I guess that'll be a tricky diagnosis to make.
Perhaps it also depends, how many people have looked at Ehrlichiosis in dingoes first. Yep.
Yeah, well,
Okay.
well, of course, before, I mean, because dingoes don't occur anywhere else in the world, we didn't have ehrlichiosis here. There's probably no one's looked at it at all yet. But we'll see. Maybe captive dingoes might get infected, and we'll hear more about it.
Thank you. Liz Tudor has a question. How might you expect the disease presentation to change as Ehrlichia becomes endemic?
Yeah, that's a great question, Liz. Thank you, and hello. Well, I think it will settle into an endemic stability, for want of a better word. And if you look at the literature, and if you talk to vets overseas, where the disease is endemic, they will invariably tell you, oh yes, look, it's quite a serious disease, but most dogs survive it, or we treat it with doxycycline and they get better. And I'm sure that a similar thing will occur in Australia over time. Unfortunately, we're just going to have to live with it. But what has been so extraordinary is this combination of the ticks and the dogs and the situation in the northern parts of Australia that I think have only really ever been seen once before and well described, as I said, during the Vietnam War. But it will become a widespread disease that all vets will have to have on the differential diagnosis list. They'll have to have it in the back of their mind. And as I said answer to a previous question, dogs with strange hematological abnormalities or fever of unknown origin, those sorts of things, then vector-borne diseases should always be on the list, and differential diagnosis list, and Ehrlichia now becomes more prominent.
Great, thank you. Any other question for Peter? If no other question, I would like to thank you, Peter, for a fantastic talk and also sparing time. We are really grateful for that, and hope to see you soon at some ESP conferences or something like that.
Thank you very much. Lovely to give the talk. Thank you very much for attending, and great to see so many familiar names and people I used to know. Thanks very much, everybody.
Take care. Thanks,
Bye.
everyone. Bye.
Bye.